Hello, thank you for joining us for this research and development discussion to take a look at our fibrosis and malaria programs. I'm Kevin Kircher, I'm the Communications Director for Ocean Biomedical, and we're lucky to be joined today by Dr. Jack Elias and Dr. Jake Kurtis. Dr. Elias is the chairman of our scientific advisory board for Ocean, and Jake Kurtis is the chief scientist for the company. Thank you both for taking time out of your busy days to come talk.
Our pleasure.
Pleasure.
Great to have you. Jack Elias is the past president of the American Association of Physicians. He's also a past physician-in-chief at Yale New Haven Hospital. He recently retired as the Dean of Medicine and Biological Sciences at Brown University here in Providence. And I think that covers probably about 1/10 of the titles you've held just in the last 10 years, maybe. Dr. Kurtis is the past director of the Center for International Health Research, and he's executive director of Brown's MD-PhD program, which puts out some of the top researchers in medicine. And he's also the current chair of the Department of Pathology and Laboratory Medicine at Brown. I can say that combined, these two gentlemen have massive decades of research experience, and in addition to whatever your personal achievements have been, I know you've run departments, helped encourage research broadly in the world. And I'll just throw it to you. The first question is, how important in the 21st century, how important is it to have cutting-edge scientific research? How big a time is this for medicine?
I'll go first. It's a very, very exciting time for medicine. The kinds of discoveries that are being made now are the basic building blocks of the mechanisms of disease. Once you know the mechanisms of disease, you can then say, how do I control that? How do I augment it if it's a beneficial mechanism? How do I blunt it if it's a pathologic mechanism? And we're on the verge of having treatments for diseases that we have for 20 years not had treatments for. And we're on the verge of being able to treat patients effectively for diseases that when I was in medical school and when Jake was in medical school were diseases that we learned did not have treatments at all.
Yeah, totally. I think if you look at Jack's career, if you were to define the sort of oeuvre, it was putting translational science and translational medicine forward. And, I can think of, coming from the infectious disease space, I can think of no greater example of that than our recent history with the COVID pandemic, where we went from new pathogen, never seen before on the planet, to understanding what the disease process was, to an effective vaccine in people's arms in 6 to 9 months. I mean, truly remarkable. So I think this is a ripe time to harvest the fruits of our basic science work that's been going on for decades and translating it into patients.
Terrific. Today we are planning to take a look at 2 of Ocean's 3 current programs, fibrosis program and the malaria program. We had talked about putting cancer in here as well, and there's so much exciting stuff going on. It's so big. We decided to hold that hopefully for another day, but and dig in on malaria and fibrosis. And I want to ask the two of you first. Ocean Biomedical, you formed this company a few years ago, and one of the principles that was founded on was helping to bridge the translational gap from discovery, bench in the lab, to bedside. And I'm wondering, and also you've stated a number of times, to meet large unmet medical needs. And I want to know, for each of your programs, how do those goals fit into your program? And Jack, I'll ask you first, why now in this day and age, why fibrosis? Why should we be worried about fibrosis? Why are you focused on it to the degree that you are?
Happy to answer. When you're talking about fibrosis, you're talking about tissue in the body that is designed to do something special, whether it's the lung for breathing, whether it's the liver for metabolism, and so on. And those areas, those tissues in the human body get replaced by scar tissue, and scar tissue does not have the same effect that the specialized tissue does. We now know that about 50% of the morbidity and mortality in diseases of visceral organs, those are the organs inside of our body, about 50% of it is due to the pathologic accumulation of scar tissue. And we now know that if we can control that scar tissue, we can have major effects on those diseases. We're talking focusing initially on idiopathic pulmonary fibrosis and a genetic disease called Hermansky-Pudlak syndrome, but you're also talking about cirrhosis of the liver, whether it's alcoholic or from some other mechanism. We're also talking about diseases like scleroderma. When you put them all together, there are a large number of people who suffer because fibrosis cannot be controlled properly in their body, and as a result, they have diseases that really cause all sorts of difficulties.
Interesting. Jake, how about for you? I don't think anyone has worried about malaria in the United States for at least 90 years.
Yep.
Why should we be worried about this? Why has this been a focus of your career?
That's a great question. So malaria is the greatest single agent killer of children on the planet, bar none. Last year, 627,000 individuals died of malaria. Roughly 90% of them were in children in sub-Saharan Africa. There are 2 to 3 billion individuals on the planet live in areas where malaria is transmitted. And there were 200 to 300 million infections last year. So it is a giant portion of the total burden of disease on the planet. So for that reason, we're really excited to be working on both vaccine programs, therapeutic antibody programs, and small molecule drug programs.
Jake, do you want to comment on people who travel to Africa?
Oh, absolutely. Yeah, so there's another market there for sure that needs protection. And, right now, the death rate from malaria is rising, in part due to our best drugs at the moment, the artemisinin family of compounds. We now have parasites with high-grade resistance to those drugs in Southeast Asia, and they've actually been detected in sub-Saharan Africa. And we know what's going to happen next. The death rate will rise as drug-resistant parasites spread. So travelers to those areas need protection, and individuals living in those areas need protection. And as you guys know in the recent news, we now have malaria autochthonous spread, so real spread of malaria here in the United States in 2 or 3. I don't want to put too sharp a point on it, but malaria will come back in areas where it has either been eliminated or it's never been as climate change continues to progress. So we do have to be aware of it.
I'm seeing we had a question come in about the current malaria vaccine. There was a lot of press over the last year that a malaria vaccine had come out. Do we need another one?
Yeah, so I would encourage individuals who are interested in that vaccine to read the phase 3 study that was published several years ago. What you'll see is it has a 10.6% efficacy against severe malaria after 3 doses, which rises to 16% after 4 doses. That, believe it or not, is slightly less effective than a bed net. So it's an important proof of concept, but it is by no means a panacea. We have lots of disease and death still occurring despite that vaccine. Those of us in the field who've been following the development of the vaccine for the last 30 years expected this result. And so we have always viewed this as an important proof of principle, but we clearly have to do better. Let me put it into a context that maybe our viewers will be more familiar with. How would you have felt if your COVID vaccine protected you from severe disease by 10%? Do you think that would have encouraged vaccination?
Do you have any idea what the actual efficacy was of the COVID vaccine?
Oh,
by comparison?
upwards on the, depending on which metric you use, it's in the 90s for the lipid encapsulated mRNA vaccines. So really amazingly effective, and more effective at preventing severe disease and infection, as we all knew. So yeah, it really apples and oranges in terms of efficacy.
All right. And it sounds like a lot of good progress has been made in terms of malaria, but this company's hoping to do better.
Yeah, we clearly need better vaccines. And new approaches, which I think we'll talk about in a little bit, but new approaches to identifying. How do you know what the right target is? The vaccine that's on the market, the CSP-based vaccine, that was identified in a mouse model of malaria. And it does really well at protecting mice from getting mouse malaria. It's just not that great for humans.
Jack, I want to ask you, how did you make your antifibrosis discovery? What's unique about it that has caused a bit of a stir?
So it's been known for quite a while that a molecule called transforming growth factor beta particularly transforming growth factor beta 1, is a major driver of fibrosis. People then spent a large amount of time trying to figure out how to control the effects of TGF-beta. We discovered that a different molecule called chitinase 1 was required for fibrosis to be induced by transforming growth factor beta. So we're in a situation where if we could control chitinase 1, we would potentially be able to control TGF-beta and control fibrosis downstream. We were lucky at the time. We were in New Haven at Yale, and the Yale University acquired the what is now called the West Campus of Yale. It was the Bayer pharmaceutical site, and they left a compound library of 6,000 compounds for Yale to look at. We developed an assay and screened 6,000 compounds, and we came up with a number of hits, and the most exciting one to us is a molecule, moiety called kasugamycin. Kasugamycin was initially developed as an antibiotic, but it turns out it's not a real great antibiotic, but it's a great antifibrotic. It's already been in man, and it's known to be safe and effective in many settings, and is used commonly in the agrochemical arena. So we have a moiety that can control fibrosis via a very powerful pathway that is easy to make and safe and effective.
We've had a number of conversations over the years about this, and one of the most compelling things I remember hearing from you is your frustration as a pulmonologist with the existing treatments that are out there for pulmonary fibrosis. And I was just wondering, can you say a little bit about that? What is the current state of available treatments out there? What are the current patient needs? When
I started as a pulmonologist interested in pulmonary fibrosis, there were really no effective therapies at all. I remember very vividly the meeting of the American Thoracic Society where the first drug that was shown to be effective for pulmonary fibrosis, for idiopathic pulmonary fibrosis, got FDA approval, and people were almost in tears that we finally had the understanding that we could tackle this disease and make treatments for this disease that would prove to be effective. We now have 2 FDA-approved drugs. One is called pirfenidone, and the other is called nintedanib, and they work through different mechanisms. They're effective to some degree, but they are not the kind of drugs that turn the disease off. They are not disease modifying, and unfortunately, both of them have significant side effects, which limits their use in many, many people.
All right, Jay, can you tell me what is the discovery that you made that caused so many waves with science, and you got your publication in Nature? What is it about your malaria vaccine program that's different than different and special?
I think it's a great question. I think it's a sort of 3 issues. One, we approach the problem completely differently. So the standard approach to identifying a malaria vaccine was to begin with mouse models of malaria. And the problem is mice don't get human malaria, they get mouse malaria. And mouse malaria is not that similar to human malaria. So instead of starting with mouse models, we actually started in the field. We follow large cohorts of children over time in endemic areas, and we figure out which children develop resistance and which children don't develop resistance. And then we ask the question, what immunologically is different in the antibody profile of a resistant child, compare it to a susceptible child? And that difference between the two, those are actionable vaccine targets. If we could make the susceptible kids make antibodies like the resistant kids do, they would be resistant. And so, we figured out sort of a genetic gymnastics approach to answering the question, what's different in the antibody profile of resistant susceptible kid? And 2, the target we identified, one of them is called GARP, is really unusual. It's on the surface of the red cell, antibody binds to it, and that causes the parasite to die. That had never been described before, and I think that's what got us into Nature. And then, of course, the efficacy that we see in our non-human primates. We're already in non-human primates, we've done them. And you vaccinate monkeys with non-human primates on the lipid encapsulated mRNA platform, the exact same platform used by the Pfizer-BioNTech COVID vaccine. The reason for that is I got lucky and met Drew Weissman in 2017 before he was famous. He was one of the architects of that vaccine delivery platform, and he convinced me to move our malaria assets onto his platform. So I think it was, we start with humans, we identified a target that had remarkable efficacy, and we've moved it onto lipid encapsulated mRNA and gone into monkeys. I think those are the three things that combined, I think, have really driven our program.
As a layperson, it sounds to me like this is a naturally occurring mechanism to kill the parasite. Is that fair to say?
It's one of the things that distinguish. We started with humans, children that had developed resistance. So they do it naturally. Not every child will die of malaria. We're interested in what differentiates the children who develop resistance. What is special about them? And can we co-opt it as a vaccine? So yes, we are building on Mother Nature, so to speak. We're building on natural acquired immunity.
Interesting. You mentioned monkey studies. We've had a couple questions basically about how far along the studies are for each of these programs. So I want to ask you a couple questions about that. And maybe Jake would start there. What have been the steps in terms of your research program? How far have you gotten?
Yeah, so the malaria program really is three programs in one. It is a lipid encapsulated mRNA-based vaccine. It's a therapeutic antibody, and it's a small molecule drug. So I'll just talk about the lipid encapsulated mRNA vaccine. So that vaccine has been trialed in mouse models for immunogenicity. So we know it's immunogenic. It's been trialed in mouse models for efficacy, so surrogate markers of efficacy, where you vaccinate the mouse, and then you take the mouse sera, and you see if the antibodies in that sera kill parasites. We know they kill parasites. And it's been in a non-human primate model of efficacy, where you vaccinate monkeys, and then you challenge them with real-life falciparum malaria, the kind of malaria that would kill any of us in the room. You challenge the monkeys with real-life falciparum malaria, and you follow the monkeys. And the vaccinated monkeys control their infection and self-cure, and the unvaccinated monkeys, they actually get very high parasitemias, and we have to drug treat them. So we've been all the way through the non-human primates. We're ready for GMP production, IND filing, and phase 1 studies.
At each of those stages, what kind of results did you see? Have they been consistent as you get more sophisticated in the studies? What levels?
So vaccinated mice, if you vaccinate mice with GARP as a recombinant protein, which is sort of a classic, old, in my opinion, old style vaccine approach. If you vaccinate them with recombinant protein, DNA vaccine constructs, or a lipid encapsulated mRNA, you generate antibodies that bind to GARP on the surface of infected red cells and kill parasites. So the actual modality of delivery is less important than the target itself. We get 99, we actually fight about whether there's a live parasite underneath the microscope.
In vitro.
Yeah, exactly. I mean, it's 99% of these parasites die, which is fantastic.
In the mouse models.
That's vaccinating mice.
Okay.
Then you take the serum and put it on parasites and culture. In our monkey studies, our monkeys self-cure. So they get parasitemia, and then the parasitemia resolves. And that, if you could replicate that in children in sub-Saharan Africa, you would have a profound impact on morbidity and mortality of this disease.
Amazing. Jack, how about for you? What steps have you taken the antifibrosis testing through? Where are you on that? What kind of results have you seen?
So the antifibrosis approach has two components to it. One is moving kasugamycin forward as an antifibrotic, and the other is then using medicinal chemistry approaches to get kasugamycin modifications that make it even more powerful and more useful. Right now, we are focusing initially on IND enabling studies for a kasugamycin to use it in the way it is produced now. We know that it's easily produced, and we know that it's effective whether you're giving it systemically, whether you're giving it orally, and otherwise. So we're at a point where we're moving down the road toward having appropriate INDs certification. And then it's going to be ready to take into man with in idiopathic pulmonary fibrosis or Hermansky-Pudlak syndrome.
What kind of results did you see in the trials that you did? And how many different types of mouse trials have been done?
So one of the things that got people in the fibrosis world saddened for many years was that there were a number of potential drugs, but when they tested them in one model, they would work. And then if they tested them in a different model, they would not work. And so we sort of said, we're not going to get interested in anything as an antifibrotic to take into man unless it works in every antifibrotic model that we can come up with. And so we now have 4 different models that we use to look at the antifibrotic efficacy of an agent. We have models where we take driver genes such as transforming growth factor beta and make genetically modified mice and target their lungs with TGF beta, and they get tremendously exciting pulmonary fibrosis. We have models where we give toxins to the lungs of mice. One of the toxins that's used most commonly is called bleomycin, and that gives a very impressive fibrotic response. And there is a model called the pale ear mouse. And it's a funny name, but it's not a funny reason to be using it. The pale ear mouse has the identical genetic mutations that humans with Hermansky-Pudlak syndrome have. And they have a massively heightened sensitivity to develop pulmonary fibrosis. So we've been able to test kasugamycin in that setting, and that is the first animal model of any sort where it's been shown that the fibrosis in the pale ear mouse can be controlled. In this case, it's controlled by kasugamycin.
And in those different models, how much consistency are you getting in the effect? And
in those models, we get about an 85 to 95% decrease in scar tissue accumulation in the lungs, no matter which model we use. It's tremendously powerful, and it really changes the outcome for those mice, and hopefully we'll have similar profound effects in humans once we take it into the human diseases.
Terrific. Here's a couple of questions for you from, I believe, from an institutional investor. Sent these over. The first one is, when do you envision obtaining initial approvals from the FDA to begin trials for each program? Jack, do you want to?
Well, I'm assuming with kasugamycin, because it's already been in man and because it's been extensively studied by the EPA, that we're talking 8 to 12 months kind of a thing before we have IND and are able to go forward. That to me would be a reasonable amount of time and an exciting outcome. We're working very hard to get that to happen.
And Jake, how about you?
Yeah, so similar timeline. What's been really important for, I never want to say something, the silver lining of COVID, but the silver lining of COVID was 2 things from my point of view. One, the clear, overwhelming safety data for lipid encapsulated mRNA vaccines, where it really is the safest vaccine delivery platform ever created. So we have that. And 2, the GMP manufacturing capacity for lipid encapsulated mRNA. Neither one of those 2 things existed before COVID, and now they're clear roadmaps for moving into IND. So again, we're looking at 8 to 12 months.
And I'll just say, I know that those are both your personal ambitious goals. It depends on
Many
factors beyond your control. Yes. The same investor also asked, which program is further along? And maybe
Mine.
I'm kidding.
Okay.
Totally kidding.
And maybe we can put cancer in the mix on this too. But what do you think?
It depends on what you mean by further along. The cancer program, one of the fundamental parts of that program, which we're not going to get into detail on today, is the development of an antibody against a molecule called chitinase 3-like 1 And the antibody that we've generated there and have tested is tremendously effective. And it's so effective that we've already sent it to a company that's going to move it along from a manufacturing scale-up point of view and give us the GMP studies, the GMP material that we need to do those studies. So you could argue that that antibody is furthest along. But once we get that antibody, we then have to put it into trials in people with cancer, and that requires a lot of time, a lot of energy, and a lot of effort. And there are lots of competing therapies that will be trying to get into the same patient populations. On the fibrosis side, you could also argue that fibrosis, that kasugamycin is most advanced, because kasugamycin has already been in man. In fact, it was in man in the 1960s as an antibiotic trying to treat Pseudomonas urinary tract infections. They never moved it forward because it wasn't a very good antibiotic, but they had no reason to look for its ability to control fibrosis. So that is another place where I think we're well down the road. On the other hand, Jake will tell you that he's got the infrastructure that he needs to do his clinical trials, which is a tremendously exciting thing. So let me pass it to Jake to talk about the amazing infrastructure that he has for the malaria clinical trial.
One of the sort of oddities of malaria is, when I speak with US-based scientists or physicians, they're like, "Oh, it must be so hard to do those clinical trials. You got to, go to endemic countries." Turns out, one advantage, it's sort of a perverse advantage of malaria, is the infection pressure is so high. So in the area that I've been working in for the past literally 24 years, in Western Kenya, it's a little town called Kisumu. We work just outside of Kisumu. The biting rate of infectious mosquitoes is insane. Every kid gets bitten by one infectious mosquito every single day. So when I go into a community at the start of a high transmission season, and I look at 2 to 7-year-olds, it's a cohort that we're currently running, I look at 2 to 7-year-olds, cross-sectionally, half of them have malaria on that day. And if I treat that whole cohort of 300 or 400 kids, and I follow them every week with a blood film to measure more malaria when they get reinfected, half of them are reinfected in 6 weeks. And so what's remarkable is you can get efficacy endpoints in one transmission season. And the high transmission season I'm specifically referring to, the March transmission season, it lasts about 16 to 18 weeks. And so you'll have efficacy endpoints in 18 weeks following your first round of vaccinations.
And you have some degree of infrastructure set up to potentially plug into?
We've been running observational longitudinal studies there literally for 24 years. So yes, absolutely. It's actually it's really an incredible field site. Physicians, scientists, laboratory technicians, field workers, really the whole package.
Another question that I'm seeing is how long you envision the trial for malaria and fibrosis lasting, and maybe you just answered that.
3 months of vaccination followed by a high transmission season.
Okay.
The fibrosis is going to be a little bit different, and it will probably be different in idiopathic pulmonary fibrosis versus Hermansky-Pudlak syndrome. But in some ways, we're very lucky. We know what the time course was for the drugs that are presently FDA approved. We're talking in the range of 72 weeks for the clinical trial, for the efficacy clinical trial. And I'm going to guess we'll probably be in the same ballpark for idiopathic pulmonary fibrosis. For the studies of Hermansky-Pudlak syndrome, we're probably going to have to do them in an area of Puerto Rico where this disease seems to be endemic. There's an area in northwest Puerto Rico where Hermansky-Pudlak syndrome is actually rather common. And we have to get people who do not have pulmonary fibrosis and then treat them and see if we're able to prevent them from developing pulmonary fibrosis. And we're going to need to figure that out, and it may take a little more than 72 weeks in that setting. But it's a disease with no treatment right now, and the people who have it have formed their own support groups, and they're dying to have therapies to take into their patient populations that have a chance of working.
You've mentioned Hermansky-Pudlak syndrome a couple of times. Do you want to take that one step further and just describe
Sure.
what the disease progression is
So
for folks? When does it set in? What do they experience?
Hermansky-Pudlak syndrome is a genetic disease. There are 9 different subtypes. There may even be an additional one being described very recently. And it's a defect of what are called lysosome-related organelles. I don't expect anybody here, maybe other than him, he'll get a quiz at the end of the at the end of the session to know what they are. But it's a defect in the cell of something that needs to travel properly in the cell and then leave the cell to do what it's supposed to do. People with Hermansky-Pudlak syndrome type 1 and type 4 get spontaneous pulmonary fibrosis, and that is a major cause of morbidity and mortality for these people. These people do not respond to the drugs that we presently have for idiopathic pulmonary fibrosis. Believe me, people have tried those drugs in them, and they have not had the success that you would have loved to have seen for these people. So you've got a group of people where you know what the genetics are, you know where there's a real pocket of disease density, and now we're just dying to find a drug that actually works in this setting.
And just to make sure we make this connection, you talked before about the palliard mouse model. What is the connection between palliard mouse model and Hermansky-Pudlak?
The palliard mouse model has the same genetic mutation as HPS 1 and 4.
Okay.
So it is genetically identical to the human disease. So if we control it in the pale ear mouse model, we have a very good chance that whatever that intervention is, of that intervention working in the people with this disease.
Terrific. This is a question that popped into my mind earlier when you were talking about all the different conditions that could be potentially treated by kasugamycin. How is it possible that such a broad range of diseases could be potential targets for your one drug?
Well, we study a group of genes called the 18-glycosyl hydrolases. They come in 2 basic flavors, if you will. There are some that are true enzymes. What that means is that they make metabolic and other things happen from a biologic point of view. And then there are others that look very much like them, but they're not enzymes, and they bind to a receptor and they trigger downstream events. What we have found is that these genes are part of a fundamental process that we all have in us, where they're designed to control injury and drive repair. And when you have them and they work properly and they're appropriately adjusted, you get good results and people are healthy. The problem is they frequently are produced excessively or at the wrong time or in the wrong place, and that's when they contribute to disease.
I was just going to amplify on it. For many diseases pass through these pathways, and that's why his drug is going to be effective and has been shown to be effective in multiple forms of disease. Many diseases result in fibrosis. So if you control fibrosis, you control this. You basically control the pathways of multiple disease entities.
And even though to me it's amazing because it's internal lung issues, it's skin issues, it's liver.
It's fundamental repair from injury, no matter what the injury is the same, no matter where it is occurring.
And it's got to be appropriately controlled,
Right.
or you don't get the benefits of the repair, and you get detriments from turning that process on in the wrong setting or at the wrong level of intensity.
Yeah, here's an easy example: burns. When you get a burn, if you're lucky, you get nice scar tissue that's, it's never the same, but it's not heaped up and mottled looking. If you have exuberant scar, we call it a keloid, that's a problem, and it can actually reduce function. So there's another example where fibrosis is out of control, not properly controlled, where kasugamycin would have impact.
Interesting. Do you want to say anything about the fact that it's so interesting that we hear about cancer as a general term? There's a lot of, we hear about mental illness. I've never heard about fibrosis as a category of illness before I met you. And do you want to say anything about that? I know you've been working with it your whole life.
Well, it's funny that you have that perspective because you're not the only one. Fibrosis for many years was sort of the poor cousin of the disease world. And one of the reasons was we had no treatment for it. So for a doctor to really push a patient to diagnose a disease that there's no treatment for is a little bit of a problem, particularly if there are biopsies involved and other things as well. But as we've moved along and understood the mechanisms behind the disease in more detail, and as we've come to a place where we say, yes, we can develop drugs that are effective for this disease, people are now going back and looking at a whole variety of different diseases and realizing that fibrosis is part of the mechanism of the pathology and the morbidity and mortality in those diseases. And so the vision you had from a while ago is now being morphed into an understanding that these are diseases that we should be able to treat in the next 5, 10, 15 years.
Great, thank you. Here is a question, bit pointed. Big Pharma has a bias that you can't make money on malaria. Are they right? And is there an economic motivation for you to develop a product? I think it means, is there any economic motivation to develop a product against malaria?
Yeah, so the first thing you have to realize is the market size is ginormous. As I said in the intro, 2 to 3 billion individuals are living in areas at risk of malaria. That's the market size. That's 2 to 3 billion with a B. That's the market size for an effective vaccine. 200 to 300 million infections a year, that's the market size for an effective therapeutic. Think small molecule drug. And then, huge death rate, 5 to 600,000 a year. That's the impetus to get busy and moving with our therapies. So if you look at how vaccines are delivered to lower middle income countries right now, and we'll pick just sub-Saharan Africa, they're delivered through a program of the WHO called the EPI program, the Expanded Program in Immunizations. About 10, 12 years ago, EPI added a vaccine, the hepatitis B vaccine. Incredibly effective vaccine for a disease that does not kill children. It actually causes morbidity and mortality considerably later in life. And yet, if you vaccinate early, you'll prevent that. That vaccine costs $5 a dose. It's deemed, because of the benefit later in life, it is deemed cost effective and is supported by the EPI. Imagine now a malaria vaccine that reduces malarial mortality, let's just say by 20% or 30% or 40%. Suddenly now the impact, the cost effectiveness of that vaccine is huge and would have a huge impact on the socioeconomic status in endemic countries. And so I do believe there is a role to be played for a private-public partnership through organizations like EPI to make a malaria vaccine cost-effective and, in fact, profitable.
Can I ask you to comment on how the military might use
Oh, it's huge. Yeah, so there are several market segments. One is endemic countries, and that's EPI is going to drive that. The second are travelers and the military. So up until recently, and maybe it's still true, the U.S. Army and Navy were the second largest investors in malaria vaccine development because they realized, during Vietnam and during 71 or 72, a third of the troops actually had malaria or were recovering from malaria. So it has a huge impact on force preparation. So military market, definitely a large market. U.S. traveler market, large market. And of course, you'll have differential pricing across each of those markets.
And I know as all these reports have started coming out over the past year and a half about growing resistance, I know you've been very concerned about the fact that there is no reserve treatment for severe malaria.
Insane to me. So the principle that Kevin's bringing up is this notion of one of the fundamental tenets of antibiotic stewardship. So infectious disease docs hoard and save and protect the latest and greatest antibiotic. I call it jokingly gorillacillin. You can't get gorillacillin to treat a simple skin infection. You have to have life-threatening infections to get gorillacillin. And that's so that bacteria don't develop resistance to gorillacillin. It's called antibiotic stewardship, and it is one of the fundamental planks of how we operate in the developed world for antibiotics. No such program exists for malaria. Our best antimalarial for severe malaria is the exact same drug that you can get in what's called a duka or a small shop without a prescription, just walking in. And so what that breeds is a huge amount of drug pressure and pressure for resistance. And so what we see is we now have high-grade resistance to our best antimalarial, artemisinin, occurring. Once artemisinin is not effective, there's not a lot left in the pharmacopoeia. We're out. And so we desperately need new antimalarials, and we desperately need to have antimalarials reserved for severe malaria. And that's part of what our drug, we haven't talked about our small molecule drug program, but we actually now have small molecule drugs that kill drug-resistant parasites at low nanomolar concentration. We're really excited about that.
Yeah, tell me about that. We talked a lot about the vaccine. How did your vaccine work spin off the drug work?
Yeah, so antibody binds GARP, kills parasite. Antibody doesn't need anything else. It doesn't need cells. It doesn't need proteins in the serum called complement. Just binding alone. Binding alone kills parasites. That's a poster child for what we call a small molecule screen, where you set up an assay, you put immobilized GARP protein on a bead, you throw an antibody, it binds. What if you throw small molecule drugs in first, wash, and then see if the antibody will still stick? If you inhibit antibody binding, it's because the small molecule bound in the same place. You have identified small molecules that bind in the same place as your killer antibody. You've enriched for small molecules that will kill parasites. So we did that. We screened literally 30 million compounds. And we have two main classes right now that kill parasites at very low nanomolar concentration, without toxicity in mice at high doses, without toxicity to human cells at over 1,000-fold the dose that kills parasites, doesn't hurt the human cell. And that's in multiple human cell types, including a model of the placenta. I was asked by a reviewer, why do you care about the placenta? Because you're going to be treating pregnant women. You've got to care about the placenta. And so we're pretty excited, as you can tell. Getting all fired up about it. We're pretty excited about that. So imagine a world where there's a new antimalarial in a new class. I didn't tell you this. This drug kills parasites that are resistant to artemisinin. How cool is that?
That's amazing.
Yeah.
And it sounds like it is, and that is why we call it a malaria program, right, and not just a malaria vaccine program. The last thing I'll ask for both of you, Ocean Biomedical says it's a company that's founded to meet large unmet medical needs. You have both spent your careers helping many researchers advance their work, set up their work. You see how the discovery process works. You see how the translation process works. How important is it to each of you to make these programs successful, and then to take that next step in the Ocean Biomedical promise of helping other scientists make their translations, make their discoveries translate successfully as well?
Yeah, you go first.
Sure. All right. Well, the bottom line is, if you talk to most disease-focused scientists, whether they're physician scientists or PhD scientists, or even investigators at different levels of expertise and training, we all dream of having an impact. We all dream of developing drugs that can change the world by treating disease and making the world a better place for people who get disease. When I was the dean of the medical school, we spent a lot of time talking about the fact that there are diseases we couldn't treat 30 years ago and we still can't treat them now, and that's such a sad circumstance for the patient. But it's also a sad circumstance for the physicians where they can make the diagnosis, but they don't have a treatment for it. You know, our goal is to have as our legacy that we tackled some of the biggest, most impressive diseases, and that by combining state-of-the-art investigations with state-of-the-art clinical understanding, we had a chance, and we changed the world, whether it's the treatment of malaria, the control of fibrotic responses, or the control of cancer.
I think you nailed it, Jack. One of the pillars of Ocean we haven't really gotten into is this notion that we're going to be moving these three main programs forward, but we're also going to be bringing in, as we get going, we'll be bringing in new assets. And in that process, we'll be vetting them. You know, I don't want to say this in public, but Jack is pretty much a rock star in the science world, in particular at
I paid him a lot for that.
in particular at choosing, like picking the horses that are the winners. That's what a dean does, right? You pick the best scientists. So
Best
being able
students.
to, yes, best students, exactly. Being able to pick the best science. We both have extensive experience on NIH review panels, literally thousands of grants. We're going to be picking with colleagues that have expertise in the tech transfer industry out of universities, and colleagues that have experience in biopharma, in licensing in things that have therapeutic potential. So there's really three legs on this stool in terms of vetting new science to bring new programs in. And I have to tell you, it'll be really exciting for me to be part of a team that brings in a new cure for pick it, spina bifida. Pick the disease that you want to cure. That, to me, to be part of the team that identified it and promoted it and guided it through what can be a daunting IND phase 1 process, I will feel fabulous about that contribution. And I know you will too.
Absolutely.
I feel like we could talk probably another hour if we could do it, but let's do it again sometime. This is part of we have a series of videos that we're doing. There several of the first ones are up on the YouTube page called Science Matters, where we're trying to find ways to talk in depth, talk clearly about the science behind the company. And I do hope we can do this again sometime soon. We'll focus particularly on cancer, and maybe periodically let's just come back and do it as we have time.
I'm sure we'd be thrilled. We want to thank you for inviting us.
Thanks,
we want
Scott.
to thank everybody who tuned in. And if you have questions, send us an email and we'll answer them if we can. Thank you.
Thank you, everybody.
Thanks so much.
Let's do it again soon.